So we'll give it another moment for everybody to join our webinar. Good. So let me get started. Hello and welcome to today's webinar. My name is Michael Waxman-Lenz. I'm the Chief Executive Officer of the American Technion Society. Thank you for joining us for the third in our weekly series, Live from Israel, webinars with the Technion experts. We hope that those of you who joined us for the first two webinars found them not only informative, but also inspiring. And we think they're a great way to keep connected to our Technion family. And when I say family, I mean our extended family from around the world. Registered today are Technion supporters not only from the United States, but also from Canada, Israel, Italy, Switzerland, Brazil. We believe even Japan and China. I hope that those folks in Asia are really sound asleep and they will listen to the recording. But no matter where you live, the COVID-19 virus is causing drastic changes to our everyday lives. Because life will but eventually, we believe life will go back to some version of normal. We need to be ready to meet these new challenges and work on important advances in other areas as well. So the American Technion Society is proud to be part of the Technion family and to highlight these important work that is taking place at the Technion now and in the future. And today's program will cover quite a bit of ground, and you will hear from two phenomenal academic researchers who are both academically brilliant, but also great people that you will enjoy. So to start, we'll hear from the very creative Associate Professor Noam Adir from the Technion Schulich Faculty of Chemistry, and he also holds the Bertha Hertz-Axel Chair. His research has implications in areas as diverse as green energy and medicine. Today, we'll hear him speak about his work in the development of novel antibiotics, research that also highlights the enormous interdisciplinary work and reach of the Technion. Interestingly, Professor Adir was just in New York on sabbatical. We were just hanging out together when the pandemic broke out, so you'll hear more about that. Next, after Noam Adir, you'll hear from Technion Vice President for Research and Development and External Relations, Professor Alon Wolf, for an update on what's happening on campus, both in terms of research as well as the effect of this pandemic on the campus community. So everybody who's joined, we're excited to have you. I hope that Frank and Barbara Axel, who I actually met recently in New York, have joined us, but I know that many of our supporters, Andy Wolf and Ross August and others, have joined us as well. So before we start the program, let me share, as usual, some procedural items. If you have questions for Professor Adir or for Professor Wolf during the course of this call, please use the Q&A feature that is at the bottom of your screen. So if you move the mouse down, you will see a little Q&A button. You can submit your questions through that webinar window. All questions will be held until the end of both presentations, and when my colleague Jerry Kleinman will join the call to ask some of the questions. Please note that this presentation is also recorded, and a link to that will be sent to you within the next couple of days. So Noam, it's great to see you again, and the screen, so to speak, is all yours.
Okay, well, thank you very much. It's been It was great to be there for a little while, but now we're back in Israel, and I hope that I'll be able to interest you a little bit about one of the projects that we're working on. So I guess I'll move to my presentation. Okay, I think everybody should be able to see my presentation. What I plan to do today is talk about one of the subjects that we work on in my lab, kind of briefly, without too many details, trying to give you a little bit of the flavor of not only the work, but how we kind of got to where we are. What is our thought process in trying to get something very new, something very different, and perhaps also something that will be useful in the future. And afterwards, I'd be happy to answer any questions that might arise, and that questions may arise after we've finished the webinar, and they can always be sent to me. You can get my email from the ATS. So my lab is called the Macromolecular Structure Determination Laboratory. It was established back about 25 years ago with the help of the ATS, actually the women's division. It was the first lab of structural biology at the Technion, and structural biology means that we look at how biology occurs on the molecular level, trying to understand everything through the three-dimensional structures of the molecules that perform all of the different cellular subjects and functions that have to happen. We have many different projects. Many of them have to do with determining structures, and you can see here, turning here a three-dimensional structure of a protein that we solved, and enzymes that are important for biotechnology and for medicine. About half the group works on photosynthesis, how plants, algae, and bacteria are able to take the energy of the sun and turn it into chemical energy. So we have all kinds of different projects. And as you can see, we have all kinds of very beautiful things that we create. It's a little bit of art that we work on. And within that, we've gotten into the area really of trying to do the creation of clean energy from photosynthetic systems directly without going through biofuels and other things like that. I won't talk about that today, and I'll be happy to come back some other time and talk about that. And today I want to talk about a project which is kind of on the side and came out of a kind of a crazy idea, and I hope that it will be interesting. And the idea is how can we perhaps create a completely new kind of antibiotic or antimicrobial agent. Before I really start, and as I already mentioned by Michael, we can't ignore the fact that COVID-19 is affecting all of our lives. And I just wanted to mention two projects that are not from my lab, but they are in our faculty, the Schulich Faculty of Chemistry in the Technion. And of course, attacking something like COVID-19 is not just developing a vaccine. The vaccine may be a year and a half away or may never arise. Some viruses are very difficult to attack with vaccines. And we need to use other technologies to try to get ahead of the virus. One of the projects is that of Yoav Eishen, who is actually seen here. It's pretty hard to see in the picture, but that's our president, Uri Sivan, next to him from a picture from about 25 years ago when they worked together. At the time, they were making conductive DNA. Yoav is a very talented organic chemist that works on materials, and he's developing self-sterilizing coatings for gowns. So you have a surface, a material that just is completely sterile all the time because it either can shed molecules or it can attract molecules depending on what you want it to do. And he's hoping to actually have this very quickly out into the market. And if this works, if these types of materials work, they can actually be applied using 3D printing into making parts that the hospital needs to replace, like tubing and other things that they need. So this is something that he's working on. And Professor Alon Wolf, who is over here, he's a physical chemist working on the area of magnetic resonance. He's able to monitor things like oxygen and CO2 in the lungs of patients using very, very small special magnetic sensors, and also something that will be able to determine all kinds of aspects of having the virus and the viral proteins in patients. So these are kind of two very different aspects of the type of work that is done in chemistry against the COVID-19 problem that we're all facing today. So more to what I'm talking about today, and I put it on the top here. We really don't have a choice, and I think if COVID-19 has shown us that as advanced as we are always really fighting against nature. Nature will always try to find a way to use us for their own benefit. And one of the things is bacteria. We all know that we host within us many different types of bacteria. Most of them are benevolent, and we need them for our everyday life. We have them in our stomachs, and they're very good, but we also are attacked by all kinds of bacteria that are very bad. And as we take antibiotics, which were developed less than 100 years ago, more and more, and as you know, we're all Jewish mothers, even the fathers amongst us are Jewish mothers. And so if the kid is sick, then we give them antibiotics, and that's great. But what we've created are many bacteria that are resistant to antibiotics. And the number of strains of bacteria that are becoming more and more resistant is going up. And you can see here that there's a prediction of 10 million deaths by 2050 by bacterium that are resistant to antibiotics. And we now understand these numbers. These numbers are terrible. And the problem with these bacterium also is that they are attacking people at their weakest when they're in hospitals. So we need new antibiotics all the time. And the problem is that most of our antibiotics were developed already 50 years ago. All we really are doing now is changing the same antibiotics and adding small chemical modifications in the hope that maybe we can overcome some of the resistance. And what this is creating is bacterium that are multi-resistant. That is, they're resistant to many different antibiotics. And the problem is, in the last few years, we have very, very few new antibiotics coming up. And as maybe some of you know, developing a new drug as it is very difficult. It takes sometimes 10 years and over $2 billion to get a new drug out into the market. So we have a real problem here. Where there's no question we're being attacked right now by new resistant strains of bacteria, and we don't have new antibiotics. So people all over the world are working on antibiotics. I should mention another problem is that the pharmaceutical companies aren't really interested in going into this area of development because it's not as lucrative as some other drug systems are. And so we have to also make systems that will be varied enough so that a pharmaceutical company will really take this on and develop this further and get this into the market. So how do we go about it? How do you start something that's completely new and not just add on something that already exists, like let's say a penicillin with one more group added onto it? So how do we start something new? So our idea was that we can start with evolution. And I could have started with COVID-19 because obviously the reason we're harmed now by this new, it's not a new virus. It's basically a derivative of an old virus that's been around. It didn't hurt anybody, and something happened to it, and it changed. And it didn't change for any reason. It's because it's changing all the time. That's the way evolution works. We're always changing by evolution, and it became harmful. So I thought I would give you a different example, something that's a little bit more visual. Back in the 19th century, in England, there was a moth. And this moth was usually speckled. It's called a peppered moth. And when it lands on a tree, you can barely see it. In fact, I don't know if you can see this here. There's a moth here. And it's hidden very nicely. And of course, that means that the birds are going to have a hard time finding it to eat it. And of course, the birds want to eat the moth. And if there are dark moths, because they are different genetically, then the birds will come and eat it. And since there are less dark moths to have children, we'll have mostly peppered moths. But the Industrial Revolution meant there's a lot of coal being burnt, there's a lot of smoke. The trees became covered with soot, and the lichens that gave color to the trees became blacker and blacker. And all of a sudden, all the dark moths, they started being hidden in the soot. And all the light moths, well, they became very attractive for the birds. And so very quickly, in fact, within 50 years, 98% of the moths. So you see an example here of evolution as it's happening. So our question basically was, can we use evolution in some way to understand something new about bacteria in order to develop a new antibiotic? Now, when we're talking about evolution, we have something that works for us very nicely, and that's called bioinformatics. Bioinformatics is like a flashlight. We have enormous amounts of data today in biomedicine, all kinds of things. We know all about the genes of humans and bacteria and other organisms. We know things about their proteins. We know things about all the metabolites they have in their cells. And we have these great computers, and we have great computer programs that can search these data sets if we know how to ask the right questions. We have to ask the proper questions. So we ask, can we use bioinformatics and evolution to understand something? Now, bioinformatics is like Superman because we have these great computers. So we have this super speed. It can see everything that's happening in biology, like having X-ray vision, and it can see everything. But you know that Superman has a problem. He can't see past lead, right? So what happens is that the bad guys always catch Lois Lane. They put her in a cave or in a room that's lined with lead, and he doesn't see Lois Lane. And now he's looking around for Lois Lane, but he can't see her. And trouble is that he's not really that smart, because really, if he knew anything about bioinformatics, he would understand that when you see a pattern, you also see what you don't see. That is, you see the parts of the pattern that are missing. And we said, well, can we actually look at what's missing from our data sets? Can we use something we can call reverse bioinformatics and discover things that are hidden from our eyes just by knowing everything that we can see? Can we find weaknesses in the bacteria? So now just a little bit of biology. I'm going to give you biology 101 in one slide. This is called the central dogma. All organisms, from the simplest, from COVID-19, which is not alive, it's a virus, is not really alive. It only has part of what I show you here, but all other organisms, they have all of their information in molecules called DNA, and DNA is coded in 4 letters, A, G, C, and T. And this is moved into a type of information that can be read. It's like an output, and it has 4 pieces of information also called AGCU. And this turns into proteins, and the proteins now have 20 pieces of information. And all kinds of letters, and you can see them on the screen. And each letter has a different chemical meaning. So each chemical meaning is going to give a chemical property to our protein. And proteins have anywhere between 200 and 1,000 amino acids. These are the pieces of chemical information. And the protein will now fold up. And if there's a bad piece of this protein, that is something, a little piece of amino acids that are bad for the bacterium, let's say. They're sticky. They're going to make something go bad. Let's say they're poisonous. So by folding this into a protein, this is what actually happens. Those sticky pieces get hidden in the middle, and the protein is good. Nothing happens to the bacterium. However, if it was something that was bad, evolution would get rid of it. Why would it get rid of it? Because if there's something bad, the ones that have the bad thing go away, and the ones that don't have the bad thing have more children, and then slowly but surely, evolution gets rid of anything that is bad. But there's one place that all proteins have to go through, and that's the ribosome. The ribosome is a gigantic molecule that is a machine that makes proteins. So all proteins, when they're made, go through the ribosome. In fact, they all go through the same place in the ribosome. There's this tunnel here. And by the way, we know, of course, what the ribosome looks like because of Adi Yonat from the Weizmann Institute, who got the Nobel Prize in 2009 for solving the 3-dimensional structure of the ribosome. So we know what it looks like, and we know what happens there, and we know that all the proteins have to go through the same place, unfolded. So now if there are bad sequences, they're going to get stuck there. So we said, okay, let's find the bad sequences, and if we find them, if we're smart enough to ask the questions correctly, maybe we can use them to develop a new antibiotic. So this is what we did. Now, of course, this is great to have a nice idea, but you actually have to have somebody to do the work. And luckily, I had 2 really nice graduate students in the lab that were, of course, supported by the ATS and by stipends of the ATS. And Sharon Navon is the main student that did most of the work that I'm going to describe in the first part, and Tali Schwartzman helped her. In any case, we used mathematics and computers to look through all the proteins in a bacterium called E. coli. E. coli is found in all of us, in all of our stomachs. It's a great bacterium as long as it's held in check. You don't want it in the wrong place, but as long as it's where it's supposed to be, it helps us live. And what we found immediately was that there is indeed a few sequences that should have been relatively found a lot, but they're found little. This graph is kind of a little bit counterintuitive because whatever is missing is actually high on the graph. So this sequence called CMY here should have been 7 times more than we found it. And this GPP was about 5 times less prevalent. Okay, we said, that's interesting. What about other bacterium? So if you look at Salmonella, for instance, which is also a very dangerous bacterium, we see that it also has these bad sequences, but they're different than E. coli. And if we look at Staphylococcus, they also have, we call them URSs, so I'm going to use this name, underrepresented sequences. So they also have them. So Clostridium, which is a terrible bacterium, has different ones. Let's say this FTW. And Mycoplasma, which nobody, believe me, wants to be, has a Mycoplasma infection, has different URSs. In fact, some of them never appear at all. So we thought, okay, bioinformatically, this is maybe interesting, but we're experimentalists. What do we do? Oh, one more thing. We did this for humans also. And humans don't have URSs, at least not short ones. This is good because if you want to develop a new chemical to be an antibiotic, you don't want it to be attracted to something that will harm humans. So this sounded good. So what we did is we took these little URSs and we stuck them into little proteins and we made the bacterium make the proteins. And I'm not going to go into the details, but these bars here show how much protein was made. So when we put the URSs in, the ones that didn't do anything are high, and the ones that affected the bacterium strongly are low. And some of the URSs were so strong that no protein at all was made. So we proved that our URS idea is useful, perhaps. And we were able to get a very nice paper, which was published in a very nice journal. And of course, the students got their degrees. Everything is good, but that's still not an antibiotic. Okay, so now what do we do? We did it in other proteins. We showed this in different ways. We showed that it does not harm human cells. Human cells don't care about this at all. And that was even better. So now this is already towards an antibiotic, but that's not good enough. We used a collaboration with Stanford and used a very unique method in order to map out where our sequence sticks to the ribosome, and we were able to map it in the ribosome to a certain position. So this was also very important. So now, how do we make an antibiotic? We have to do a couple things. First of all, we have to be able to get the URS inside. So we thought we would make small peptides. A peptide is just a series of amino acids that are connected to one another. They can be 3 amino acids, 4, 5, 6, or more. We started small. Then we have to get the peptides into the bacteria. By the way, it's very easy to make peptides. We can make them for very cheap. We had to find a way to get the bacteria in, the peptides into the bacteria and then get into the ribosome. And now we have to see, does it work? So I'm, of course, jumping into, we've done a lot of work here, but I don't have enough time to describe all the work. These plates are plates that we use to grow bacterium. And on the right side, on this side, and on this side, you kind of see this smear. That smear is bacterium growing happily on this side of the plate. On this other side of the plate, we put our peptide with our URS. And as you can see, it's completely transparent even after 40 hours. So the peptide went into the bacterium and it just killed them completely. Even after 40 hours, no bacterium have grown. We are also able to do this in a more sophisticated manner where we're following the growth of the bacterium over time. And if we don't do anything, the bacteria grow up. This is the typical graph which shows the growth of bacteria. The higher the line here, the more bacteria there are. If we add our bacteria, it takes them a lot of time, hours and hours and hours before they start to grow. And I'm not going to go into the math here, but this is basically, we killed about 98 or 99% of the bacterium with one application of our peptide. And of course, as you know, when you take antibiotics, you usually have to take this for many days because the first time you kill about 90% of the bacteria, you have to take more to kill 90% of those that were left, and so on and so forth. So it looked quite nice. At this point, we also started working with another very, very talented researcher at the Technion, Professor Yaron from our Faculty of Biotechnology and Food Engineering, who recruited 2 new students, Reut and Nofar. And I think I didn't mention here, this is Tarek Tarabe, who is a graduate student in my lab. And all of us together are working on this project. And here, this is Salmonella. And Salmonella, like E. coli, are also affected by our URS. Now, of course, we needed money to get this project going. And luckily, the Israeli Innovation Authority allocated a certain amount of money in a special type of project, which is called a Technion project. It's not a lot of money, but it was at least enough to kind of get this started. We also did some work to show that our peptides bind to the ribosome in the correct place. And so we're at a point now where we know we don't yet have an antibiotic, but we have a chemical, a small peptide that kills bacteria. And if we can turn this into something that a pharmaceutical company would like to now formulate and turn into something useful, we believe that this platform will be very varied. In fact, we will be able to overcome resistance, because when a bacterium becomes resistant to one URS, we're able to give it a second URS, which it doesn't have resistance to. So we'll kill 99% of our light moths with one URS, and the 1% of dark moths will kill with the second ERS. So that's our hope that we're going to be able to do in the future. And hopefully we'll be able to do something useful in the very, very near future with that. And of course, I have to say thank you to all the members of my lab. Of course, being a professor in the Technion is a privilege. One of the privileges is to teach the new generation. Not all the people in the pictures here work on the same project. We work on many different things. And I have to thank my collaborators from Stanford and from the Technion, the past members, and the Israel Innovation Authority that gave us money. And I hope that I've interested you, and I'll be happy to answer any questions that you might have. And I'll
thank you so much. Yes, thank you so much, Noam. Good. I always love listening to you. I love your energy, and I love what I learn. I have to say, maybe if I had Professor Adir in my high school or college, you know, who combined evolution, bioinformatics, and Superman, maybe I would have done something different in my life. But no, I really appreciate your insights. And obviously, we've all heard about the general challenge in the world around antibiotics, but you really make it come alive of what kind of solutions, potential solutions are being worked on. So really, thank you so much. Based on the time, I'm going to move on very quickly to Alon, but let me remind everybody, you can continue to submit questions through the Q&A feature at the bottom of the webinar screen. So please feel free to submit questions, and then Jerry Kleinman can pick them up later on. But Alon Wolf, without further ado, please, I'd love you to give us some update of what is going on at the Technion today.
Good evening, Michael. Good morning, and everyone, depending on what time zone you are. Let me also share my screen with you, so I'll show you what's going on the Technion right now. And so this is a picture taken, these are pictures taken yesterday, and as you can see, nothing much is going on campus, although everything happens now in virtual spaces. And I encourage all of you to go online and maybe read at least the promo or the description of a book by Isaac Asimov from 1957, The Naked Sun. So we are all sharing and working, but in a virtual world. So this is what's happening in Technion right now. This is the Senate building down at the bottom, and the Student Union building, for those of you who know, on the right side, and the main center alone and on the left side of the picture. However, as I said, there's a lot of things going in the background. We have many labs working currently on COVID-19 focused research and other technology to assist medical staff to fight the virus. As you can see on the left-hand side, this is the entrance to the Technion. This is the gate. So keep calm, stay at home. We are working on it 24/7, as we say. We have around 31 or 32 labs now working on COVID-19 problems, trying to solve them. On the right-hand side, you see medical staff helping a patient, and apparently this is the #1 challenge that we were asked to solve while talking with Rambam Medical Center, one of the biggest medical centers in the northern part of Israel, associated with the Technion. And it is to try to protect as much as we can the medical staff. One of the major concerns is the medical staff are being infected or exposed to COVID-19, and then when people need doctors, there is no one to take care of them. Some examples, I'll give you a few examples of research going on. This is a research by Professor Roy Kishoni. You can see him on the left-hand side on the red sweatshirt. It was all over the news. He was able to come up with methods to actually reduce the number of samples that you have to take, or the exams that you have to take in order to identify people that carry the virus, and reduce the number by a lot. And actually was using an old method that, when he actually published his ideas, everyone said, oh, we know this for 20 years. Yes, but he was smart enough to pick it up and actually do something useful with it. And it's actually being implemented now in Israel. So as I said, one of the major concern is infecting the medical staff. This is, we had about a week and a half ago, a very large, I would say, webinar and a Zoom discussion with senior administration and doctors from Rambam Medical Center about, how can we help them, engineering wise in the short term. And one of the major concerns they say is the aerosols, the spray that patients are producing when breathing is infecting them, and this is the #1 concern. So a very successful research that actually took less than a week is by Professor Eyal Zussman, a world expert on nanofibers, and he was able to produce this yellow sticker that is actually produced in his lab, and it's made from nanofibers covered with all kind of chemistry that actually, once you stick it to a conventional medical mask, it will protect the doctor or the medical staff and actually prevent the COVID-19 virus to actually penetrate or actually reach the respiration system of the doctor. This is a great success. It's actually being tested now in Nahariya Medical Center, approved by the Israeli Ministry of Health. And I'm told today that millions of stickers are already being ordered around the world, because if it works, if it actually works as it's promised, then it's a great, great success. This is Professor Ezri Tarazi, who developed again another method to try to protect the medical staff by producing a mask again. And on the right-hand side, you'll see there is a constant airflow going on this transparent glass that he had, preventing aerosols going into his towards his face, and therefore protecting him. And by the way, this was also tested in Rambam Medical Center in different departments, and also in COVID-19 departments, and it's going to be implemented probably the early next week in many medical centers around Israel. Another challenge is to develop robots to assist medical staff instead of exposing them to patients. So we addressed our first robotic teams around in Israel, because this is a project out of the Technion to build robots to try to help those medical staff, and a team from Haifa actually joined us. And this is the result you see on the left-hand side, the robot. And the person you see here on the doctor that you see here in the center is Dr. Hussein. She's in charge of the 2 COVID-19 units in Rambam now, exploring the robots. It was a great challenge to build a robot in less than a week that would serve patients, bring them food, medications, and also would be able to take measurements, external sensors to the patients without exposing, again, the medical staff. On the upper picture, you could see 2 lines, red line and yellow line. And this is how it works in hospitals. You can go all the way to the yellow line. You should not, and you cannot cross to the red line, because the red line is the infected zone. So the robot is about to do that without exposing the doctors. This is a video taken about 2 days ago. This is the new department that it was put together to actually host the patients, the new patients of COVID-19. You know, there's a big increase in Israel. And you see the robot is actually serving between the beds, moving around, controlled by the doctors. One of the main challenges was to do a very simple controller for the doctors. And there's a screen, the doctor can talk to the patients and get information. So these are some of the research that is ongoing on campus. There's, of course, Professor Hussam Hayek, who works on sniffers and stickers that can detect the virus. There is Professor Shuka Zevi, who works on using some kind of an iPhone technology and AI, artificial intelligence, and by respiration or respiratory audio, detect whether a person is infected or not. A lot is going on. We're actually putting, working 24/7 to try and help medical teams and develop all kind of therapies like Joshua Schnitzman, who is developing a liquid form therapy that enables drug delivery very efficiently to COVID-19 patients that suffer from severe acute respiratory systems. Other than that, as you could see, the campus is pretty empty. No one is allowed to get into the campus. We only allow 15% of the staff, only then what we call emergency staff, to go into campus. Those who work in the animal facilities to feed the animals, electricians, and only emergency staff and administration. All the workers, everyone works from home. And another great challenge was that in 4 days we had to switch into distance learning, like many universities around the world. Again, Zoom is probably saving us all. We have now more than 80% of our courses taught remotely, and I have one student here at home who study at Technion, and I can see how effective it is, with the exception, of course, that we cannot have labs, practical labs, done remotely. But I can tell you that the overall satisfaction of the students and the faculty is tremendous. And it raises a lot of questions, how would academia and teaching is going to look like after COVID-19. So these are challenges that we need to confront. It's a very dynamic situation. We meet, the administration, we meet on a daily basis with the deans and also between ourselves and try to tackle or deal with problems that arise on a daily basis. So this is the reality. It's a very dynamic environment right now, or situation. This is in a nutshell what is happening on campus. I'll be happy to answer more questions if you have. And the floor is yours, Michael.
Thank you so much, Alon, for giving us this picture of what is actually going on physically, but also these tidbits of really unbelievable research projects, experiments, how one can help. I mean, it's just fascinating to see. So I'd like to turn it over to the questions, and we'll invite in a moment my colleague Jerry Kleinman, who is the ATS Director and Principal for Principal Gifts. Obviously, those of you who see him on the screen, he's not in Israel. It's just his Zoom background that he's pretending, while Professor Wolf and Professor Adir are in Israel. But let me kick off the questions, maybe, Noam, where we left off in terms of what were you researching here on your sabbatical in New York, and how did the outbreak of COVID-19 affect the work you were doing?
Well, I had actually arrived on the invitation of Professor Marilyn Gunner from the physics department of CUNY on the CCNY campus, which is on the upper west side, kind of north of Columbia. Professor Gunner is an expert in electron transfer and photosynthesis. I'm an expert in energy transfer and photosynthesis. And my idea was to really get her interested in the hope that we could collaborate. She's a theoretical physicist. If anybody sees the Big Bang Theory, of course, those are the real physicists, the theoreticians. And I have been working on energy transfer and photosynthetic antennas for many years. I managed to arrive and kind of get settled, and I was there a couple of times, and I even managed to be involved in the beginning of a project that is kind of a co-project along with Yale and with Rutgers. I managed to give a group meeting there, a group meeting to Rutgers, also talked with people in Yale. And then things kind of started to close down. Columbia was closed, and then CUNY was closed, and we kind of got stuck in the apartment. And at some point we felt that not only was this not very productive for us, but also that I think that staying there would have been just adding more people to New York's problems, and we didn't think that was the right thing to do. They have enough problems without having a couple of Israelis, so we decided to go home. I continued to try to to work with Maryland from afar, and hopefully that's what's going to happen. And I haven't completely canceled the sabbatical yet, and we'll see how fast things happen. And you know, if it's not this year, then maybe it'll be next year. I don't know. We'll see.
Terry, go ahead. Thank you.
Thank you, Noam. Thank you, Alon. We have a number of questions on a number of different topics. And I guess for both of you, but maybe we'll start with Alon. And I know your background behind you doesn't show a gorilla, but someone said want to know about the 800-pound gorilla in the room. Are these labs really up and running on campus with all the mishigas going on? How do you maintain that and collaborate with other universities and other labs? And then someone else asked, what do you need to keep this moving forward quickly?
So the labs that are on campus, they have a special approval to work. It's like a matter of national security to open these labs. And actually, the collaboration is cross-country. It's not just, you know, it's Technion with the associate hospitals, it's Technion with the Ministry of Defense, it's Technion with other universities. I personally have, aside for the robotic, you know, I'm a roboticist, so we're working on the robots. I have a collaboration now with Hebrew University about designing those something which is a deformable mask that would seal completely the face of the doctors, protecting them from the aerosols. So it's a collaboration. It's an ongoing collaboration. There's constantly, as I said, it's a very, very dynamic environment where ideas are popping, and I know you just raise ideas, and you know, if there is science behind them and there is a chance that you be able to help someone, then you can actually follow your ideas and get permissions. You know, we get permissions from Rambam Medical Center to travel there and test our whatever we do. You know, we go in and out of the hospital, though, you know, well protected. So yeah, it's a crisis now, and you know, in Israel we know how to deal with crisis, unfortunately. But this time it's a global crisis. It's not just us. It's not just us in the game. Something that we did do is we actually put two. Actually, we call them emergency funds out, because all these activities, you know, we need to run them. One is to help us support all the activities related to the COVID-19. You know, Josua's research and other researchers. And of course, the help for the students who need to somehow function in these difficult times. So, but right now we don't, you know, we just do what we have to do. And as I said, it's an ongoing process, very dynamic process. Every morning you see more and more activities, more and more research going on, and research which is in the short term. I mean, not something that would result in 10 years. All we need is solutions right now. You know, respiratory machines, you know, designing them, printing something, printing materials, printing tools to doctors. So this is the dynamic. This is a very dynamic campus right now. Although if you walk outside, nothing. It looks empty, but it's very dynamic.
So
perhaps I should, one thing I should mention, however, is that the supply lines, of course, are also to a certain extent cut off. So it means that people have to be, to a certain extent, more inventive. They have to work with what they have. It's kind of like this thing where you, hey, okay, what can we do with what we have? Because you can't order anything from really anywhere easily. We have problems, all kinds of things that we were waiting for in our peptide project, which of course we can't really do because it's not connected with COVID-19. But even before the cutoff started, things started to slow down. So I think that at some point, some type of supply lines are going to have to be open in order to continue some of these projects. People are going to need things that they need in order to you know, get their ideas you know, off the board and into the hospitals.
Well, I'll give you another. It's a you know, interesting point what you raised. You know, I heard today in the news that, you know, Israel is producing now a lot of respiratory machines, and turns out that we're missing several parts. There are Israeli companies producing these, but some of the parts came from different places in the world that you cannot import now. So Israeli Air Force industry actually shut down an entire factory that was producing something completely else, for space missions. And now they are producing those missing parts to those companies. So it's a national, I would say, focus. It's really everywhere. Everyone is involved.
Several people commented about their concerns, just as their concerns for the healthcare workers in this country and elsewhere. They voiced concerns about the students and faculty members and staff still manning those labs, operating those labs.
It's a good point. We're very strict about the regulations. So those who work at the lab follow the instructions given by the Ministry of Health and the government. So no more than 5 people in a lab, and they keep a distance of at least 3 to 4 meters. And of course, they're protected, gloves and everything. So everything that is going on is not actually putting in risk the people that work there, because we do follow the regulation of the Ministry of Health. Everything is controlled. Everything is reported to the Ministry of Health on a daily basis. So they know exactly who entered the campus, and each day the list is being updated by the Director General, Professor Golani, and everything is controlled to the best of our ability and to the instructions that we get from the Ministry of Health and the government.
And as a question related to that, someone asked, are there not people who really are on campus who have no place else to go? And so they're basically on campus during this crisis.
So we have around 4,000 students and families that live on campus in the dorms. The dorms are considered a neighborhood, and practically as like a neighborhood. We did encourage, those who can actually go have some kind of a, what we call family or and other alternatives to go out of campus, but there's still quite, a few thousand, 3,000, I think already, 1,000 left, but we have also international students who are on campus. So they are in the dorms, and this is exactly, we consider this as a neighborhood, and they're not allowed to leave the neighborhood. They have their supermarket and all the services there. And if they need to leave for a pharmacy or etc., they have a special permit where they can go out and in of the gates. Now, the only way to get into campus now, if the Technion is a campus. You cannot jump. Well, you can if you want, but you can. There's a fence around it. But in order to get into the gates, we have a special permit by number. So I have to show my permit, and I have to take. They have the guards taking my temperature with this laser gun every morning to see that I don't have any fever or something like this. So we really follow the instructions. There's no shortcuts here, and everything is controlled by the instruction given to us.
Noam,
Yes.
Someone's asking, if viruses replicate, why do we keep referring to them as not living organisms?
Okay, so this really comes down to semantics to a certain extent. What is life? Okay, because anybody that likes science fiction, you start having artificial intelligence and robots. Are they alive or are they not alive? If a robot can build another robot, is he alive? So it's a little bit of semantics. It's considered by most biologists that in order to have an organism that's alive, even if it's a parasite, which means that it's an organism that lives on something that it gets from another organism, it has to have a cell cycle that is able to reproduce its own DNA, make its own proteins, enclose itself in a barrier of some type. Viruses don't have that ability. They're basically a little bit of genetic material surrounded by a little bit of protein. And all the machinery comes from the host cell. But I'm sure that this is something that we could argue, I guess, about what is life. At least when I was a student, it was considered not to be alive, and who knows? Maybe COVID-19 will make people change their idea. But that's still my feeling, is that the simplest organisms that are alive have to have the capability, the metabolism. They have to be able to uptake nutrients and transform them to energy. Viruses can't do that. They don't, unless a host gives them energy, they can't do anything. They don't make their own nucleic acids, etc. So that's why.
Well, if you wonder why I was smiling, is because I'm a roboticist, and there's an ongoing debate whether, you know, and you mentioned robots. Are they human, or are they alive, or are they whatever? But maybe this is a topic for a different webinar.
Is your faculty appointment in the Faculty of Health Sciences or the medical school? That's very interesting. So I have a financial question, but it's for Professor Adir.
Yes.
So why do we need a bank for proteins, and how do scientists make deposits and withdrawals?
Okay. So that's an interesting question, actually. About, I would say, 40 years ago, when there was an explosion of the number of people creating these protein structures, that the community got together and said, look, we need to standardize how we do what we do and how we check that what we're doing is correct. And by the foresight of governments, especially the United States, Japan, and the European Union, the Protein Data Bank was formed in order to enable all of the world to benefit from the three-dimensional structures. I should mention that today, in order to really develop a new drug, one of the prerequisites is to know what is the target. It's no more magic. We want to develop a molecule which binds to a certain biomolecule, and we have to understand what it does. And the three-dimensional structures is what gives us the ability to map out how these things interact. So the Protein Data Bank was established, and it's one of the rare cases where we allow somebody to actually check our data and to find mistakes. You have to understand that we're talking about thousands and thousands of atoms that are in each structure, including water molecules that are bound, because in many cases, the actual activity is due to a certain water molecule bound to a certain amino acid within a certain protein. And there are computer programs and actual people, once I deposit the structure, that will actually tell me, you know, we think that these two atoms should be a little bit further apart. And for me to actually publish the structure in a journal, I have to show them that I have deposited my protein structure in the PDB and that it has been approved. So this is very special, but it means that a pharmaceutical company can go in, doesn't have to ask any questions. By the way, we can hold on to a structure for 1 year. If we're working on, let's say, on a drug or something like that, so we have a head start. But after 1 year, it's released for the benefit of mankind. So it's a bank that thinks about mankind and not only about its own.
I got it. I think I got it. Alon, somebody asked a question about Professor Zussman's, I believe that's the way it's pronounced, his patch material, yes. And maybe I lost it here. They want to know if it was resistant to the virus. Is that what this is all about?
Well, it's first is the nanofiber. You know, he's using electrospinning to create those nanofibers. He's done it for many years now. You know, they even use it for, you know, those fabrics that when you go into airports, when we used to go into airports, they took this to look for if there is a gunpowder leftovers on your suitcases. So he was working on that things. And it's the nanofiber structure and the material that the nanofibers are made of and covered with, that both block the size of the virus. The virus cannot just, you know, it's like a filter. The virus cannot go through this sticker, and the material there also destroys the virus. So it does two things: block and destroy.
Are there impediments? Someone else says, are there impediments other than finances for moving these inventions and IP forward? I mean, there's a couple dozen on campus. Does Technion have what it needs, or are there impediments that Israeli society and government and corporate use has to hold these things back?
So of course, everything which is used in medical facilities has to go through, you know, all the regulations, etc. But everything goes very fast now. So I'll give you the example of the robot, for example, because, you know, I've been involved with this. So I can give you. So one thing is the reliability, the communication. You need to communicate with the robot. You don't want the robot to get stuck or hit someone someone walking in the department there, and sterilization. It has to go through very strict sterilization, and so everything has to go through, there are conventional regulations, but things are more relaxed now. I would say that we can do this very fast. There are fast track committees that approve these technologies that you see an immediate effect, like those stickers. They were approved in a couple of days by committee. It was tested. Now a medical center is testing them out. If all goes well, it's going to be spread out to the entire country. So we are in an emergency. Israelis, work, as I said, unfortunately good when there is time pressure. We need to improvise, but still keeping all the regulation as much as we can and not taking any shortcuts. I doubt it that any medication that is new, completely new, unknown, would be approved in no time just to someone to swallow it. But this is the mode of operation. And as I said, if we're, the challenges are mainly that, as Noam said, you're missing materials, parts, and then there are I would say officials in the country where you can actually apply and say, hey, we need this and we need that, and they're going to find those missing parts and materials for you. Actually, yesterday it was interesting. It was in the news that the Mossad is also involved in getting all kind of. It was on the TV, on public TV. So I don't think I'm telling any state secrets here, and that they actually were looking for all those sticker tools or what do you call this?
Ventilators?
No. To measure whether someone is carrying the virus. No, how do you call it? This in Hebrew? I know I don't know.
Swab or the test or the.
Yeah. They were calling or mask or yeah. So it's really what do you call it?
Long Q-tips.
Yeah. So it's a national. Everyone is involved.
So one last question, Noam. Are we going to be facing COVID-20?
Excuse me?
Is COVID-19 going to change to COVID-20 and we're going to be back where we are next fall?
Well, of course, I'm not a prophet, but SARS was in 2002 and MERS was about 5, 6, 7 years ago. Both of them are from the same family. Yes, I imagine that there will be a variant, probably not in 1 year, but modern life has made us been able to transfer diseases very quickly in the world. So I would not be surprised, but maybe we'll learn something from this on how to behave when, certainly the biggest thing is international cooperation on notification about the appearance of something that looks different than what has already been seen, and working together. Now, the trouble is, of course, that countries are still not really working together. We all read the news. We know that some countries are working differently than other countries. But in order to avoid something like this, if they don't work quickly together, then yes, I'm afraid that it's going to happen.
That's sobering. Michael, I'll turn it over to you.
I was going to say, I was hoping you would start on launch or end on an upbeat note, the sobering thought. But I do really want to thank both of you for spending your evening with us. It's always fascinating. For a moment, I was afraid you would discuss the meaning of life, but you stuck with what is life. So I really appreciate your insights. And I actually think, from my perspective, this webinar and many of the other efforts show, while this is a global virus, there's also, I feel like, a global pulling together. I hope that translates into finding solutions and that there is good collaboration. I feel like, as I see with the Technion family, as I said at the beginning, there's a real pulling together and an emotional support. So thank you for joining. For those who are on this webinar, we have recorded the presentation. We will share it with you. And we look forward to having you join us again. next Tuesday, April 7th at 12 p.m. Eastern Standard Time, 11 Central Time, 9 a.m. Pacific Standard Time, and any other time zone that you may be. And you will get to hear Technion Assistant Professor Yaron Fuchs, who heads the Laboratory of Stem Cell Biology and Regenerative Medicine in the Faculty of Biology. And he will talk about the intriguing topic of cell death, the key to regrowth of fingers and combating the coronavirus. You should receive an invitation to this upcoming speaker event live from Israel. And if not, just come to the ats.org website. Thank you so much for joining. Stay safe, stay healthy. Wish all the best to your families, and thank you for your support for the Technion. Thank you to everybody. This ends this webinar.